Our first speaker is Dr. Sidney Brenner. Dr. Sidney Brenner really doesn't require much of introduction at all, and it is our utmost pleasure and honor to have him in this session as a speaker. For those unlucky few who do not know Sidney Brenner or do need introduction, I advise you to Google Sidney Brenner, founder of molecular biology, mRNA, roundworm, and Nobel Prize for that matter. Okay, let's invite Sidney Brenner to the microphone.
Well, when I was asked to come and talk at this meeting, I found I could fit it in on a stop between Japan and Singapore. But I could only come for my talk. So I came last night and I'm leaving this evening. I apologize for that. It would have been interesting not only to stay for longer, but also to see many, many old friends here. So I was asked, what will be the title of your talk? It's just same as the last one. And that is, I'm going to talk about genes, basically. But I also have some things to say about the subject of your meeting, which is called synthetic biology, which I have to admit is a terrible name. In fact, its only virtue at the moment is, and let us all pray for it, that it'll replace systems biology. It's okay. So I don't know what the word synthetic biology means, but I can tell you what people outside think it might mean, which is the creation of monsters. And I personally, although I think that my interests overlap fairly considerably with what most of you are engaged in, I don't call it that. I call it molecular engineering. And I think that's a very good name. And I want to say that, and I thought I invented this. And so I looked it up, and I discovered that it was invented by our old friend. And if you care to look in the Proceedings of the National Academy in 1982, you will find an article entitled Molecular Engineering, written by Drexler, the same man who invented nanotechnology, and it says exactly what you want to do. You know, it has exactly the same phraseology. If we can design the sequences of genes, we will be able to make proteins, we will be able to do everything. Okay, so I think it was all done 1982. It's a quarter of a century ago. So conceptually, that has been there all the time. So what is different, I've asked myself, about synthetic biology and synthetic anything, right? And I think this concept is quite important, but why I call it molecular engineering is to take advantage of what we can do in engineering in order to create what we want to create in the future. So many years I wrote an article about centaurs. I don't know if any of you have read it. It's a very short article. So as you know, a centaur is, the bottom part is a horse, Okay, and the top part is a man. There doesn't appear to have been any female centaurs. So unclear how they reproduce, but that is okay. So the question I posed is, in future, for an examination in synthetic biology, which is the answer you would have to do is it's produce the genome which would produce a centaur. Okay, now it's quite difficult when you come to think of it, because what the Greeks did, the ancient Greeks, is they cut off the top part of a man and just joined it to a horse. That's easy. But what you begin to realize is this is now a hexapod. It's got 6 limbs. It has the arms of a man. It has the forelegs of a horse. It has the rear legs of a horse. Okay? It has 2 chests. So it's got 2 hearts. So, and 2 ways of breathing, but it's not clear how you're going to connect these 2. I mean, you're going to ride the hearts in tandem. You're going to inactivate one. So when you begin to think about mythical animals, which is what I suppose is the aim of doing this, and and for Chinese synthetic biology, we need to make a dragon. We need to make a dragon, and we're going to have some problems with the flames, I can tell you that. I can tell you that straight away. And I don't know, you see, I don't think if you look at not only mythical animals, but you look also at allegorical animals and so on, where people weren't even sure what kind of this. And if you look at the Chinese lion has dog's feet. Doesn't have cat's feet, because the lion is a cat, not a dog. So the question is, the Chinese must have had a way of reprogramming dog DNA so that it gave us cat feet. That would be an interesting exercise for novices to see how they would do it. Now, of course, when it all boils down to things, I think we can ask ourselves what it is we want to do with this. And I will give you my view of it, because it's very different, not very different from this. In fact, I was told that after this meeting, everybody will say, he is a synthetic biologist, but he doesn't know he's one. You see? So I'll tell you what I think you have to abstract. What are the essential features of the biological system that we want to get hold of? Okay? We know a lot about design and construction in the field of physics and chemistry. I call it chemistry, but it's actually another form of physics. But with the world, the material world there, we know a lot about how to put things together. We know a lot how to make devices from them. We know a lot about their construction. And we've actually succeeded in making very complex objects, like the aeroplane many of us use to fly here, which has hundreds and thousands of components, and still seem to work, basically. So we know that, and that's the product of engineering. We know how to design things under constraints, and we know how to test them, and we're still learning a lot about how to put things together and do the regulation of them. Of course, and that proceeds by knowing something about what are the constraints of natural materials, what you can do with them, and of course, by us being engineers. That is, by making a design which in principle will work, and being able to implement it and test it. And the art, indeed, of engineering consists of that. But let's look at this other group of complex things that are natural, which is the animate world. And let's ask how they have achieved complexity. It has been achieved in a completely different way, not by design, but by a process of evolution constrained by natural selection. All right? And I think we have to ask the question whether we want to give that up as an engineering principle. That is the great power of the great power of not solving things uniquely, but looking at things and just taking the things that work. Because that's nature, right? Indeed, mathematics is the art of the perfect, and physics is the art of the optimal, and biology is the art of the satisfactory. If it works, we'll use it. So don't worry about looking for elegant solutions or optimal solution. It just worked, and in fact, I once thought I would start a new journal. It was going to be called JCC, the Journal of the Crumbling Cookie. It was a journal about evolution, and you would only be allowed one line of the discussion. And the discussion line was, that's the way the cookie crumbled. That's the way it happened. So we have another powerful natural process in operation which we yet barely understand. And there's still a lot to be done on that, and a lot of things to learn from that which we can do. Now, of course, there is one area which demands, I think, immediate attention. And if you look at the biological things that are available, what you realize that they can do, which we can't yet do, is they can do exquisite self-assembly of very complex molecules. And if you will look as to how all the conditions for the optimal synthesis prevent the recombination of what is split to actually transfer things from one part to another, you see, this is all achieved by a very complex molecular assembly, just being together inside this bacteria. Okay? And which has given rise to, let's just taking photosynthesis as an example, perfected in bacteria, borrowed by plants. That is, they engulfed the bacteria and tamed them to use them to generate this activity. But there's one very big difference between the things we make and the things biological systems make, and that is durability. Okay? Biological system, things are not durable. They fall apart. Why? Because it's cheap to make them again. I mean, you don't have to make an enzyme that is going to last for, 20 years without being renewed. Enzyme, if it's, you just make some more of it. Right? So there's been no natural selection necessarily for durability. Actually, since everything started by the need to develop life in an environment very different from ours, roughly speaking, an environment that was essentially boiling water, no oxygen, okay, and just smelt of hydrogen sulfide all the time. That is, the primitive soup wasn't a soup, I can tell you that. So that one way of looking for these very ancient organisms is to find conditions that still exist today, which are equivalent to the early paleo environment, and see if there's still organisms there, because they will be wonderful fossils. They are the wonderful biological fossils of the past. And I don't have to tell you that there are thermophiles that do astonishing chemistry, that can do it in boiling water, that there are things which can exist on salt, on sulfur, use sulfur as the terminal electron acceptor, and that everything else has sort of been a kind of decadency from that. In other words, once a lot of oxygen came around, you could move to, more decorative things, all right? So we can then ask ourselves the question, are there any other ways of making biological systems durable, right? So my view is this: I see biological things as amazing fabrication. They can fabricate, so I don't see them as anything else, right? And what I would like to do, and in fact one of the things I want to do, is I want to do what the plants did. I want to make durable chloroplasts, capture them, okay, and make artificial leaves that we could just cut out with scissors. That would be, and I think that's achievable. It's achievable. But this raises very interesting questions, which is, how would we not only achieve that, that can be achieved by various ways of taking the fabricators. It's possible to destroy most of what they've got, just leave them with the bare minimum, make that stable in various ways. One could polymerize them once you have them in some kind of plastic environment, so that you would stop all the other things happening. And there are many, many technologies there, but the one thing, once you start to do this, and anybody who has started to think of devices which use biological things like biosensors will realize that the interface between the biological and the physical is very primitive. Usually consists in sort of, charging it up with lysine and sort of shooting the DNA in it, hoping it sticks. It ranges from that to other things, but the actual communication of it is something that we would like, that needs improvement. And that is going to take molecular engineering. How can we not only make the parts that we take out of bacteria durable, but that we can actually integrate them in other devices? How can you make DNA talk to a computer more directly and not go through some kind of imaging with a very poor sort of output, a very low resolution output, low information content, because it's very hard to get more than 6 lines in the visible spectrum. that are orthogonal to each other. So we need a lot of that. And so I believe that before we can design the mythical centaur, I think that there is an opportunity to do many things, and of course I have to just tell you that of course many of these things are being done and will be done. But I want to tell you some amusing stories now of my attempt to be a synthetic biologist in the year about before Drexel wrote that. So the year I would say is about 1980. I decided that all this wonderful new stuff we could do, cloning genes and so on, could be put to use. And I discovered by reading in the literature, I was interested in nematodes, that a wonderful compound had been found from the fermentation of streptomycetes called avermectin. Avermectin is one of these antibiotics, a very large macrocyclic ring. I was absolutely killed nematodes, stone dead, and didn't have any effect on mammals. It turns out that now we know it is an inhibitor of one of the neural receptors. It doesn't kill us because it doesn't pass the blood-brain barrier. Okay, so I thought, now, what do we want to do? So I conceived of the idea of cloning the genes from the streptomycete and putting them into rhizobium. Well, I still was smart enough then to realize it's too difficult to put them into a plant. But I'll put them into rhizobium, which is, you know, is a symbiotic bacteria, and then I could infect the plants, and these are in the roots, the nodules there, and that's where the nematodes get in and eat the plants. So I would develop this biocontrol reagent. And I actually got funding to do this from a company which doesn't exist anymore. It was called, one of the numerous ones, called AgriGenetics, gave me a grant. And I actually started by trying to clone the genes from streptomyces. So, of course, in those days, you needed a probe. So how do you clone these genes? Nobody knew. Right? So I said, look, the way this compound is built up, we knew a bit about the biosynthesis, as I think this is a modification of fatty acid biosynthesis, where you pass the thing around and you extend the chain. And I said, this will be an enzyme that will have many of the characteristics. So why don't we make probes against the sequence? We have the amino acid sequence. Of course, we didn't have many of the genes at those stage, but we have the amino acid, and we'll try and clone this gene with degenerate probes. All right? And so that is how we started. In those days, you took this and you made an extract of the DNA, and we did gels, and we did it, and we actually got to something which, when we sequenced it, looked a bit like this. However, this is when the money ran out. I think the company ran down. Okay, and we were out. But the project was one that I think you see required something which is beyond, was really beyond our capacity at the time. And only now, I mean, we can now do this now. We've cloned all these genes from these. People are making recombinant proteins which make new antibiotics. We can stick it into this. So that was too premature. But we need to think, and what the interesting thing about that was that didn't want to engineer the plant, but just something that was working in the plant to do this. That I could, of course, not only get into plants, but I could actually have something to sell. I could sell you bacteria to infect your seed, when you did this, and so you could have a nematode-resistant plant, but it wasn't forever. It wasn't for eternity. Okay, that is, I wasn't altering the plants. If you felt like you'd like your plants to be demolished by nematodes, because you were sympathetic to nematodes, you could just withhold the bacteria and let them be eaten. So that's fine. So basically, the other project I want to tell you about is the following. I got the idea we should try and make hydrogen. As you know, this is very common in what I call the biofools industry. Yes, I have to tell you my solution to all of this, and I'll tell it to you at the end. So, we could make hydrogen. We could do lots of things with it. And I discovered a lot of things that if you take bacteria that are anaerobic photosynthetic bacteria, the red and purple bacteria, that in the absence of oxygen, and in the absence of a source of nitrogen, the nitrogen-fixing bacteria can actually reduce protons. It can reduce protons to gaseous hydrogen. So we conceived of this entire idea of, but you need, of course, a carbon source as a supply of energy. So we thought this would be really great. You see, in the daytime, when the sun is shining, you accumulate the energy. And in the nighttime, when the sun goes off, you use that energy and turn it into hydrogen. So you have a complete cycle to do this. This was the idea. And indeed, since you need a lot of area to do this, we decided we would do this down in the sea, because it's very easy to convert area with optic fibers into cylindrical arrays. All right, so we would do this in things that were 500 foot long. The sun would be piped in during the day. Then hydrogen would be taken out. Okay, and then did some calculations. So what you would get out of this. And just looking at this, we found we couldn't make enough energy, actually, to make the plastic we should need to construct this. It just isn't enough, to do that. So I think we have to, when we're thinking of biofuel and all of these other things, I think you better learn some economics very quickly. You see, because I had an idea seeing the movies where, you see on an oil field, they flare off the methane and burn it. So I thought, my God, if I can find a use for methane, okay, I'll get it for nothing. So I had this idea. I discovered you could grow bacteria on methane as a carbon source. So I had a friend who was the research director for the Shell, a big oil company, and I went to see him, and I said, I have this great idea, and he let me talk to some economists there. And they said, Dr. Brenner, you may be right, but if you find a use for it, it'll have a price. And the price will be such that your final product will not compete excessively with the others. Right? Finally, at the time, together with these economists, we worked out a different plan to convert methane into biomass. And that was the following. It turns out that there was an excessive tanker capacity. Because there were lots of tankers. And in particular, there were tankers that came through the Mediterranean, went to Louisiana, and carried oil from, of course, the Middle East. But they returned empty. Okay, so our idea was we empty out the oil, we line this. We were going to do this with oil, actually, but we lined the oil tanker with plastic, so things don't get very smelly. We stop in Cuba, where there was an excess of molasses from the sugar cane. We fill this up, we put in yeast, and we take a nice slow drive back to Europe at the optimum temperature of the yeast. We come to Rotterdam, we just empty out the yeast, pick up some more oil, and do the same thing. I give you that story just to show that all of these things require not only the science and the technology to implement them, but we have to be conscious of the value of these and what it is meant to replace. And now, as I finish, I will tell you in the last 2 minutes my solution to biofuels. I think there's a hell of a lot of biofuel just walking around, especially in America, which is the fattest country in the world, and also in Australia, which is the second fattest country in the world. So my idea is very simple. We merely declare obesity to be a crime. And it's a crime to be fat. This means you can be arrested and you'll be sentenced to a place where you will find a bicycle. This bicycle will be connected to a pump, and you'll pump water up a hill where there's a big dam. All right? And you see, we can solve the health problems of the country for the same money. And of course, when your weight is reduced and so on, you will be released, but only on parole. You have to report to a police station every month to be weighed. And if you go back again, back you go. Now, of course, it's not realistic, but we're asking now to do other things which are going to be much more complicated than that. So I think in all of these areas, we should be careful that one thing we propose, which is to make biofuels from food, which is one of the projects, and the other thing is, which I think is interesting, is to ask whether you can use waste to make this. And of course, there are many projects to use cellulose to make this. And we should wait to see. And I think what you should also realize is that, of course, we don't want to run evolution again, because it took a hell of a lot of time to do. There's an old story of Saul Spiegelman, who was a very famous scientist of the old days, who was the first to do in vitro evolution with a little RNA virus. He set that up. It was beautiful experiments. This is in the 60s. And he gave his paper and showed how he could evolve the sequence of this virus just by the principles of natural selection. and this was beautiful paper. And some wit from the audience said to him, "Hey, Saul, what's new? It's been done before by God. It's called evolution." Saul's instant reply was, "Big deal. Took him 2 billion years, took us 6 months."
Thank you, Sydney. We are running a little bit late on time, but maybe a few questions would be nice.
Do you remember what happened at the end of this lecture? Somebody mentioned to Saul, your talk would be just as good as if your fly would have been closed.
I don't remember that. I think maybe you're mixing up another thing. No, I don't remember that.
Audrey Stevens said at the end.
I can't remember that. That was at Oak Ridge. It was an Oak Ridge meeting. Good. Okay.
Well, thank you very much.
We're going to do that. Good.